that we have been talking about for so long live. So my name is Alex Schofield. I'm the Associate Chair of Graduate Architecture, co-director of the Architectural Ecologies Lab.
Negor Kalantar.
Margaret Pettit. So the three of us are directors of the Architecture Ecologies Lab. So we have collaborated.
Yes.
Or over here.
Yeah.
And we'll talk about more.
Yeah. So we want to invite President of the CCA, David House, to come and initiate the day for us. Thank you, David, for being here. We are not going to introduce you. I think everybody knows you. So this is.
Good morning, everyone. Good morning, everyone. Some of you know that I like call and response, so to know that you're alive and well. But welcome to CCA. I am David House. I'm the president here at California College of the Arts, and it's a great pleasure to welcome you to this symposium, which I understand has been growing and will continue to grow over the next several years. Today, we stand at the intersection of creativity, technology, and environmental responsibility. This symposium is more than a presentation of ideas. It is an exploration of possibilities. At CCA, design is not only about form, but also about care, responsibility, and regeneration. The prototypes that you've seen or will see on display made by our students and others really embody the boldness of an inquiry and the courage to experiment. And I had the privilege of walking through 2 days ago with Nagar, and it's a really incredible exhibition, and we hope that everyone will have a chance to see it here in this room, but also tell all of your 100 friends to come. It's really a special place here at CCA, and our students are really empowered and emboldened by an incredible faculty, and I wanted just to take a moment to again recognize the organizers of today's or this symposium, Nagar, as well as Margaret and Evan and Alex. Thank you for your leadership and for bringing us all together here. Of course, they were assisted by many students and alumni, and so we welcome you all as well. This event marks an exciting new chapter at CCA because I understand we envision the symposium as a biennial tradition, and what better place to host than here in the Bay Area on CCA's campus. Speaking of new chapters, I would be remiss if I didn't acknowledge an announcement that was made today in the spirit of collaboration and partnership. We just announced this morning that there was a significant gift that was given to the college by Lori and Jensen Fong Foundation in the amount of $20 million, which was matched by an additional $20 million. No small undertaking, a $40 million gift that we announced today, which ensures that CCA continues, and it also demonstrates the power of partnerships, not only philanthropic partnerships, but industry partnerships, as well as academic and community partnerships. So I'm super excited today. And also I have to say that Lori was here on campus 2 days ago and spent about 2 hours with us walking through the campus, but got lost in the Biomaterials exhibition and did not want to leave. And at the end of our lunch, she said the only bad thing about this was she didn't get to spend enough time. She is an engineer, and she totally was having a great time there, so we gave her a parting gift. So thank you, Nagar, for walking us through and also acknowledging Dean Keith over here for his leadership as well. So to all of our speakers and panelists and students and guests, thank you for being part of this journey here at CCA and with this Biomaterials Symposium. May the conversation begin, inspire new questions, spark new collaborations, and propel us toward a more sustainable, equitable, and imaginative future where more people from different backgrounds are creating the future that we all want to inhabit together. So thank you for being here. Have a great symposium. I hope I will be walking in and out throughout the day, but I'm really thrilled to see you. And again, thank you for joining us. So I'll turn it over to Keith now.
Thank you, David. I was just sharing with President House that one of my nightmares is speaking after him. So bear with me, please. Good morning. I'm Keith Crumity, Dean of the Architecture Division at CCA. Welcome and thanks for joining us for the second day of the Pioneering Biomaterials Symposium. At CCA, we begin our events with a land acknowledgement, recognizing it as a meaningful act that acknowledges our presence on native lands and encourages reflection on our role in ongoing colonial legacies. CCA's campus, one campus now, is located in Yelaman, also known as San Francisco, on the unceded territories of the Ramaytush Ohlone peoples, who have lived on this land since time immemorial. We acknowledge the historic injustices inflicted upon Indigenous peoples, including their forced removal from ancestral lands and the systematic destruction of their communities and culture. CCA honors Indigenous peoples past, present, and future, and we wish to pay our respects to local elders. This symposium is a direct reflection of the culture of collaboration and experimental making that defines CCA architecture. Our division is committed to excellence and innovation in architecture and architectural education, advancing design as a social, ecological, and technological practice. With 4 distinctive programs, Bachelor of Architecture, Master of Architecture, Master of Advanced Architectural Design, and BFA in Interior Design, we engage students in a rigorous and forward-thinking education grounded in deep disciplinary and cultural awareness that embraces emerging technologies, material research, and digital fabrication. Our work is deeply rooted in social and ecological impact, with a commitment to community engagement, equity, and environmental responsibility. Our 4 research and teaching labs, 2 of which are responsible for why we're here today, Architectural Ecologies Lab, Urban Works Agency, Digital Craft Lab, and History Theory Experiments, serve as platforms for interdisciplinary inquiry, thinking, and making. This year, we are excited to introduce a new track in our MAD program, the Architectural Ecologies track, which will further expand our work at the intersection of design, ecology, and biomaterial innovation. This symposium is a natural extension of the values that guide the division, bringing together designers, researchers, and industry leaders to push the boundaries of experimental making and sustainable practices. The symposium underscores the power of collaboration beyond the college, bringing together diverse expertise to explore the future of biomaterials. It's been made possible through our ongoing alliance with Autodesk, established in 2021, through the great efforts of Margaret Akeda and Nagar Kalantar, working diligently with Rick Rendell and Athena Moore at Autodesk. And it also reflects our partnership ongoing with UCSF research scientists and PhD candidates. It bridges education, industry, and scientific discovery to advance material innovation. We encourage and invite all of you to collaborate with us in advancing this dialogue, fostering innovation, and shaping a more sustainable, interdisciplinary, and just future. We're immensely grateful to Autodesk for the generous support and to all of those who contributed to the exhibition and the symposium. As David mentioned, and as the lab directors themselves mentioned, the event was thoughtfully organized by Margaret, Alex, and Evan of the AEL and Nagar of the DCL. I'm going to start using acronyms now. Their leadership, along with the invaluable contributions of current CCA architecture students, Nagar, you were supposed to give me, maybe you did and I missed it. Here we are. Their leadership, along with the invaluable contributions of current CCA architecture students, Kira Finnegan, Don Lawrence, Fong Tran, and Andrew Rosum, as well as recent MAD alumnus, Ali Farjmandi, and BARC alum, Claire Leffler, has been instrumental in bringing the symposium and exhibition to life. We deeply appreciate their dedication and vision. At CCA, and you can see some of our partners in the exhibition and contributors to the exhibition. At CCA, we believe that learning is most powerful when it's hands-on through making, testing, remaking, reimagining, iterating, and doing it all over again. This symposium is not just a conversation. It's about equipping our students and emerging designers, and I would say the larger community of designers, researchers, and makers in the Bay and beyond, with the tools, knowledge, and inspiration to shape the future of biomaterials and therefore the future of our environment. We invite you to continue joining us on our expanded campus for lectures and events that explore the intersection of architecture, design, and innovation. Our next event will be held on March 11th, featuring Studio Ahead, a local design practice. You can also find details about this and other events at scaffold.architecture.cca.edu. Now I'd like to turn it over to Margaret and Nagar, who will introduce today's events. Thank you again for being here. Thank you, President House, for being here and joining us. We look forward to the conversations ahead.
Well, thank you everyone for being here. I just wanted to quickly talk about 5 years ago. So this is a culmination of something that began 5 years ago. Nagar and I booked a meeting with Rick, and we had an idea. We said, we dropped an idea about a deeper alliance with Autodesk Research. And he listened, and he graciously received us. And then there was a moment in the conversation, I saw it in his eyes, a spark went off, and he's like, if we can make this happen, we've got something here. And that was the beginning of us figuring out if we can make something happen, talking to Keith, talking to different folks to see if we could create a pipeline that could benefit both Autodesk and CCA. So that was 5 years ago during the pandemic that we had it. We started with the symposium that is built, and it built with our collaboration with UCSF. So they're our neighbors, and by having Autodesk research, industry leaders, and scientists from UCSF, really in regards to Dyke Mullins that will be part of this first panel, this kind of tripod really helped us launch something that really has grown into get the ball rolling that has allowed us to be here today. So it's something that's grown over 5 years, and we're so eternally grateful.
Yeah.
Nagar's going to talk
Yeah.
more about it.
Yes, and some other points. I want to start with a few points that I wrote down from Jenny Sabian last night as a keynote that I believe the same, so I thought that it's good to wrap up. Was the power of design, and I think that all of us that we are here, we have some connection with the design. And design is not just look, it's the period that 80% of environmental impact of anything is made at that state. So that shows the power of us, people that are in the room. So that was one of the points. The other one was the collaboration. Collaboration and synergy, it means that two people become more than two, And that's the correct collaboration. There are students in this room that maybe they said that we don't want to work in team. It means that you didn't have a right team member so far. Otherwise, I have Anna that I've worked with her for, 10 years. We have Doug, we have Autodesk people here that we work no matter what is the distance between us, because when you find the right collaborator, you stick together and work together. And then you don't need to be expert in every field. I think that was really good point to students, that you just need to do your work and communicate well. And that's how we can connect together, and that was really important. I want to add few more, that power of ask. Yes. And the first time, I felt that when Steve Jobs mentioned that I never called anybody and they didn't answer me or try to help me. And I think that's the power of, asking and sharing that this is my vision. People try to help, and I think that's the really thing for collaboration is make the, ask and people help you. And with that, I want to wrap up, what we learned from the first day, and and I want to appreciate the collaboration with Autodesk and collaboration with UCSF. It helped us to become a really we feel a synergy, and it's really crucial to have them. Today we have Atina Moore with us, that she's the Network Strategy Manager of Autodesk Research, where she leads the Autodesk Research Residency Program. In this role, and through her volunteer work in cycling, art, social justice, she has built a strong commitment that support and empower one another. She has been with Autodesk since 2015 and is based in Boston. We are deeply grateful to have you here, and I want to invite you to come and have some.
Well, I just want to start by saying +1 to everything that Margaret and Nagar said. I am beyond thrilled to be here, particularly for day 2 of Pioneering Biomaterials. As Nagar mentioned, even just last night's keynote from Jenny Sabian was so inspiring, and I found myself just in the few hours that we were together having these immediate aha conversations. So I'm so excited that we have this entire day together. I think I want to give just a little bit of context for what Autodesk and Autodesk Research do. What's this? Oh, next slide. Thank you. Oh, one more. No? Just kidding. This is where we want it to be. That's great. So yeah, just to give a little bit of context, I think, for the Alliance itself and also for Autodesk. Autodesk's mission is to empower everyone everywhere to be able to design and make anything. And the reasoning behind that is really so that we can design and make a better world for all. Autodesk Research is driving that mission by looking at where there may be opportunities where others would see roadblocks. So we're doing this, Autodesk software, our technology is spanning almost every industry. We work in architecture, engineering, and construction, product design and manufacturing, media and entertainment. So Autodesk Research is really looking across all of those industries to investigate and contextualize where our technology can play a role in supporting those emerging design and make challenges in our industries. We're also working, with our collaborators and with our customers to inspire them to think about and see these preferable futures. One of those being, net zero buildings, which are supported by something like biomaterials. This is a critical conversation. We're also, collaboration is a key kind of element of this entire event. We're working with partners who are helping us build our collective understanding of the world, our industries, the challenges, the opportunities, so that we're really prepared for this future. So one of the kind of key tools that we have in Autodesk Research to foster this collaboration is our Autodesk Research Residency Program, which myself and my team are responsible for managing, which is an honor. And in this program, we're bringing in and working together with innovators directly to kind of co-explore and co-create what is happening in our industries. Again, we're seeing those immediate challenges and opportunities as they're coming up for us. This program brings in a really diverse global array of teams from industry, academia, of which CCA is obviously one, and the startup community. So there's a lot of variety in there. And that program is actually where our relationship with CCA started, but it very quickly emerged from there. I think we could see the value. We're similarly open-minded, and so it was very interesting to take this new and kind of nebulous idea of what does that mean for us to work more deeply together and form this alliance. So for us, that's been a true partnership where we're making actionable that idea of like co-creating, co-exploring, being very experimental, presenting ourselves with some really interesting challenges that it's sometimes clear, or unclear I should say, how are we actually going to do that? And we've been very successful in doing that, so that's very encouraging for us. I think, despite the fact that Autodesk is working across all of these industries, we have our kind of eyes on a lot of different areas. We have a lot of different kinds of teams in our program that provides a lot of breadth. But even with all of that, it's really easy to kind of work in a silo, to have your head down, be working on the problem at hand. And I think that happens naturally. It's sort of a reflection of the sector that we're in, the way we understand working. And so having a partnership like CCA, like UCSF, is really pushing us to, again, consider how we break down those silos, how we blend and blur the lines, how we mesh our ideas together. And it's really exciting to continually find these ways where we can bridge areas like industry and academia to really look at what the future is. And again, build that collective knowledge, that collective resource. So it's super exciting to find those different resources and point them at a problem together, which is what we're really doing here today. So again, super excited to be here. So thankful for everyone. I know you're going to hear that a million times, but there's no small feat to have all these presenters, exhibitors, panelists, organizers, students, everyone that's been involved. So thank you so much. And with that, I'm going to hand it back to Alex.
All right, I'll keep it short and sweet. I'm just going to give us a run of show. So you were all here at the Pioneering Biomaterials Academia and Industry Convergence. And so our run of show today is as follows. In case it wasn't clear, I'll paint a picture for you. We're trying to thread a needle all the way from academia to industry. There are many people who here today who are amazing practitioners in this field. There'll probably also be people that are coming wondering, what do we mean by biomaterials? Aren't there a lot of biomaterials out there in the world? And so hopefully today we can answer some of these questions. Not all of them will have complete answers, but hopefully we'll plant a lot of seeds for further conversation as we move on to this future altogether. So we're currently just doing the welcome. Panel 1 of academia will go from 10:30 to noon. We'll have a brief break from noon to 1, which will break for lunch. And then panel 2 for convergence, sandwiched in the middle, will run from 1 to 2:30, and this will be a mix of academia and industry together, our Venn diagram. And panel 3, the industry, will run from 3 to 4:30. We will be having our keynote speaker, Dr. Lynn J. Rothschild. I was gonna say today, unfortunately, David Benjamin got COVID and could not join us. However, I'm instead going to say, fortunately, we have Lynn to speak with us. And Lynn is just an amazing individual coming from NASA Ames as a research scientist, also a professor of many courses as well, adjunct at Stanford. And so I can't say I have favorites of all the people we talked to, but man, this conversation really planted so many seeds to see some of the things that we sometimes see in academia, even industry, and kind of question, what are we doing? Why are we doing this? Is this really going to turn into something that we can actually use in our day-to-day lives? But these people are sending mushrooms to Mars. Pretty wild. So please stick around. Should be a lot of fun to really hit the nail on the head in what we're trying to do to converge all these together. So with that said, I'm going to introduce Margaret, who will introduce Camille.
We're giving our secret notes. Anyway, I wanted to introduce our first moderator for the academia panel. It's Camille Moore. Camille, we had the pleasure of working with Camille all last year. She's a PhD student, graduate student at UCSF, so she's one of 3 that worked with our team of 4. It was a large team, it's 7 of you, so 4 CCA architecture students and 3 graduate students. And so she'll talk more, but it was, it really is the power of, they are the bio, we are the design, and that merging has really resulted in some beautiful things that you'll see upstairs. So thank you for moderating this.
Of course. Yeah, thanks, Margaret. Hi, everybody. Yeah, so as Margaret said, I'm Camille, and I was part of the biodesign team last year. I'm also a PhD student at UCSF, and I'm a joint student, so these are my 2 labs. And just briefly, at UCSF, what I'm studying is really how your DNA is packaged in your nuclei. So you have about 6 feet of DNA per cell, and a cell is smaller than a grain of sand. And so this is a really complex organizational problem because the right parts of your DNA need to be accessed at certain times. So I study the biophysical properties of that DNA and proteins that then modify the biophysical properties. So the biodesign project was very different in some ways, but also similar. We were totally thinking about biophysical properties of our material and how to measure that, working with the architects at CCA. So it was a really wonderful experience. Is that it? Okay. And so then I think the panelists are going to introduce themselves. Maybe the panelists can come up. Laura, Jenny. Okay, so then we'll start with Laura. Okay, great.
Should I? Oh, right here. Perfect. Okay. Does that work? It works now. Okay, great.
Hi everyone, my name is Laura Maria Gonzalez. I'm a designer and researcher. My work bridges fields of design, computation, synthetic biology, all to explore this very broad category of biomaterials, which we're going to be diving into today. My background, like many of you perhaps in the room, is in architecture. I did my
Is the
bachelor's.
microphone working? Sorry, because I barely hear.
Can you hear me?
Yes.
So I just need to go closer. Okay, so as many of you know today, my background is in architecture. I studied at Carnegie Mellon for my BArch and then went to work professionally in New York City where I worked on the cast in place concrete building. If you're ever on Broadway and 31st, I had the pleasure to work on that building. And it taught me a lot about material, it taught me a lot about the testing that goes into producing a concrete cast in place material, but it also taught me about sort of the sustainability hurdles around it. So that started a very long journey for me around biomaterials and thinking about using biology to produce materials not only for sustainability, but also for the health of our environments. So just to speak briefly on a few projects, the image that you're seeing up on the screen is the bacterial biocement bricks. So on the left is one produced with sand, on the right is one produced with soil. I've been developing this ever since my master's at MIT. Briefly speaking on the process, the main component is a bacteria. It's called Ascasturiae. It's found commonly in soils. And while it doesn't produce crystals, it induces the formation of these crystals in a local microenvironment. So I like to think about them as collaborators that can help produce biocements or cement alternatives. And so a lot of my work has been delving into producing fabrication workflows to create reproducible and larger tiles, three-dimensional tiles that address some environmental concerns. I also dabbled a bit in synthetic biology. I think the tools and the spaces are becoming accessible to designers. And for me, that's a huge opportunity for more design thinking around these topics. And so for my thesis, I was at MIT, I was looking at not only making biocemented bricks, but also perhaps using them to sense and remediate pollutants. So I worked to engineer co-cultured, let's say, multiple microbes into this material that could sense and remediate. I've also had the pleasure to work and try to create these environments for others. So at MIT, I did a lot of work to make this process accessible. I led a lot of workshops, and that continued during my time at GenSpace. So if you guys haven't heard of GenSpace, they're a community biology lab in New York City, a really wonderful space that's open to the public and teaches biotechnology to anyone that is really interested in it. So I've had the pleasure of working with them to not only make this process more accessible, but also to continue the research. What I quickly learned is you need a biology lab to work with bacteria. So I've had a wonderful opportunity with them to continue the work, and also now as a visiting lecturer at Cornell in the Design Technology Department, also for earlier to also continue some of the research on the engineered building materials front. So I'll close up by saying that I think for me, what I find most interesting about biomaterials and engineered building materials, particularly for me, is that they're alive. They start to blur the lines between organism and material. And that's a really exciting design space, right? Like, how do we design once we recognize that it's living? How do we design with biological constraints? Right? It's not just inert. There's time to consider. So I'm really excited to be a part of the panel and to talk about the challenges and also, I think, some of the opportunities in that space.
Hi, my name is Doug Collins. I'm a professor a couple of blocks over from here at the University of California in San Francisco. My lab is in the Mission Bay campus. And I am a member and now chair, for my sense, of the Department of Cellular and Molecular Pharmacology. My background, initially I was really interested in mathematics and physics. So my undergraduate degrees, I got a degree in mathematics, and because I was told I needed to do something practical, I also got a degree in electrical engineering. But I really was not that interested in that. I went back after I got my degree to my hometown in Eastern Kentucky. I'm on a farm. I thought I was going to be a genetic farmer, and realized that was a terrible idea. And went back to graduate school just again to study electrical engineering, but was not super interested in it. My first summer, after my first year as in electrical engineering graduate school, I worked for a couple of biologists, Ava Herbst and Betty Siskin. And I worked designing and building some small nanoamp current sources for stimulating rat sciatic nerves. And I got really interested in reading about this project. I'd never taken a biology course before. I just thought biology was squishy and it was all about counting bugs. And that says more about my high school education than and I got really fascinated by it. And I realized you could apply math to it. You could think about it in a way that engineers think about it. You could use some of the tools that physicists and engineers and mathematicians use to understand biological systems. So I switched to biomedical engineering. I got my PhD in bioengineering. And initially I was really interested in what biologists call cellular signaling. So how signals are generated inside cells that cause things to happen. Or how cells generate signals that are transmitted to other cells, molecules that move from one place to another. And cells can read out both the identity of that molecule or the kind of spatial pattern of that molecule or the temporal pattern of that molecule. Is it coming out in a repeating pattern? And engineers are all over that. So this is communication theory. So I got really interested in that. And then a seminal experience for me was I took a course at the Marine Biological Laboratory in Woods Hole in Massachusetts, the physiology course. And I got really interested in what is called the cytoskeleton. And the cytoskeleton is the set of polymers that give cells their shape and mechanical properties that allow them to change shape, to move, and to organize their components, and to go from being a rowdy collection of macromolecules bouncing off of each other and not able to see beyond their own hydration shells, to establishing a common identity, to become a cell, to become life itself, not to wax too poetic about it. That's what it is. And that for me, suddenly I was like, so this is where a signal, the question becomes, how does a signal turn into a structure? How does a signal turn into a functional machine that then carries out some job and then falls apart gracefully so that the components can be used to do something else? So I became interested in cellular architecture. That's basically what my lab studies. How cells establish an internal architecture that gives them their mechanical properties, that allows them to control and change shape. And shape is extremely important for cells, most cells. And to organize their intercellular spaces. So the projects in my lab, I've been at UCSF for 25 years. The projects span eukaryotic cells like us that have nuclei, biology, where how our cells kind of establish order, to bacteria. We got interested in bacteria about almost 20 years ago with a couple of very talented students who were very brave. And so we started working on, we realized that the same molecules were working in bacteria to establish order. And more recently, the third domain of life, has anyone here ever heard of the word archaea? Okay. A third domain of life, archaea, actually there are only two domains of life, really, but ask me about that later. And so we're studying across all domains of life how the molecules come together, how they self-assemble to carry out fundamental biological processes. So that's my passion. That's what I'm really interested in. And I used to, as a side note, I mourn the loss here of the little cafe, because I used to come here almost every day and have the pad thai. It was the best pad thai walking distance from UCSF. And I used to pine for some sort of interaction with architects and artists and kind of cool people coming in and out of CCA. I actually cold called and tried to email people here to see. It wasn't until I met Evan and Margaret that I was like, I found my people. There are people at CCA who are longing to kind of get together with people on the science side of things. So I'm deeply grateful for that chance encounter. So I will end there and pass it on.
Great. Good morning. Good morning. Yeah. Good. So I'm Jenny Saban. Really close.
Yeah.
So I will try and say some things that I didn't say last night. So I'm a professor of architecture at Cornell and also part of the inaugural faculty in Design Tech, which is a brand new multi-college department of design technology that cuts across multiple departments and 5 colleges. The College of Architecture, Art and Planning is the administrating college, and we're collaborating with Cornell Human Ecology, Bowers Computing and Information Science, Engineering, and Cornell Tech in New York City, which is our tech hub on Roosevelt Island. In addition to being a professor in architecture and design tech, I'm also the chair of this new department, so I wear many hats and am principal investigator in the Sabin Lab, which is where I collaborate with a number of scientists, mathematicians, engineers, biologists, physicists, and beyond, and we look at new possibilities for responsive and adaptive materials. As I mentioned last night, a primary question that drives all of the work commencing in 2005 is how might buildings and their integrated material systems behave more like organisms do, responding and adapting to their local environments. And through that, we innovate new protocols and methods for making. We work with a number of cutting-edge techniques in digital fabrication, working with robots and multi-axis 3D printing, drones. We work across scales from the nano to the macro. And then my other role is within my practice, Jenny Sabin Studio, which is a separate team. We have a separate space in downtown Ithaca. It is more cerebral, it's clean. And that's where we're able to really kind of push the boundaries of what's possible with application and to take fundamental research that has reached a certain level of maturation into public settings. And that's where I'm able to take on commissions, exhibitions, client projects, et cetera. And most importantly, to work with diverse communities in a participatory design process. So we frequently start projects with workshops, engaging with communities. I have a really exciting project right now that we won by competition in Boston, in the Roxbury neighborhood, which is a commission with the Boston Mayor's Office of Arts and Culture. And we spent 9 months doing engagement with the community there. So it's been really exciting. And I think one of the big responsibilities that we have in the context of the work that we do is being able to communicate to diverse and broad audiences. And for me, that's really where the work makes impact and comes alive, as it were. Maybe a couple of things. I started college in biochemistry. I think it was my form of protest against my parents who are artists. And then I quickly realized, like, no, this isn't the right fit, and I shifted to ceramics and became fascinated with glaze chemistry and kiln firing, gas kiln firing, and specifically salt firing and the kind of magic of that. I was fortunate to study under Akio Takamori, Doug Jeck, J.D. Walker, Patty Woroshina, Bob Sperry, went to Rome with Patty and Bob. It was unbelievable. And in my studio, and this was in the 90s, early 2000s, I sort of had a crisis of being overwhelmed by the fact that it was totally up to me as to what project I took on. And so I really longed for more collaborators. I was still trying to find that perfect love that would integrate my interests and talent within science, math, and the design arts. And so I decided to pursue my graduate work in architecture at Penn. And so that journey continued into the formation of Lab Studio, which Christina took the first installment of a course that Dr. Peter Lloyd-Jones and I taught, which was called Nonlinear Systems Biology and Design. And ironically, but there were a lot of naysayers, even colleagues and friends, saying, what are you doing, spending all this time with these biologists? And it made sense on so many levels. But flash forward 20 years later, I'm just thrilled to see this community and the impact that it's making in the way that we teach, the way that we practice, the way that we research. And the needle is shifting in terms of recognizing the value of design, bringing design to science and the rigors of science to design. And the formation of new directorates like the Technology Innovations and Partnerships Directorate, the NSF TIP, which is focused on application. That needle is suddenly shifting because if you're focusing on technological application, you need design in that space. So there are larger grant opportunities, new potential pathways for bringing people together in formal settings that 20 years ago it was so hard. So really excited to be here. I talked about this project last night, so I won't say anything. This is Polybrick 2.0, which I have former students in the audience that worked on, which is super exciting to be part of. So I will pass it to you.
Hi, you hear me well? Yes. So I am Laia Mogenso de Vila. I'm an assistant professor of architecture at Penn and director of the Luba Lab, where we look at providing matter for generative architecture. And we think, and actually, this is a book format that's going to come up in a couple of years. And when we say reviving matter, we think about it in two ways. One is practically, like making materials that are living or living-like, that can support and interact with the environment in ways that traditional materials can't. But we also look at reviving matter as a new way of thinking about material, a revived way of thinking about material in architecture. Instead of choosing a catalog of options, now we have the agency to design it. So we are advocating for that, and we do that in all the courses and the research that we undertake. What you see here below is Tegula, which is a display at the exhibit here. And we took the opportunity, and thank you for that non-trivial style, all those styles. We took that opportunity to show materials that are interactive, that are not only benign to the environment, because we take a very radical approach to what enters the doors of our lab. It's almost stuff you can eat. If you can eat it, the earth will digest it, right? So that's the premise. So this is just, it's very simple silica sand, some flax fibers, biopolymers, and a dye that changes with UVI radiation. You'll see stuff that releases smell in the exhibit and other dyes that are able to change color according to temperature. Just anecdotes, but in a way to say that that materials and only actually biomaterials or materials that are chemically engineered can tell us about the environment in ways we've never seen before, and can diagnose threat, and tell about what's coming, and tell us about what's behind and what's not. So that's the adventure we took at the exhibit. And I actually was allowed a few slides, so we can go to the next one. I'll tell a 10-year story in 6 slides. This was 2015 at MIT when we started working on the water-based fabrication platform, because we realized that architecture or material making is rarely water-based, while in biology it's mostly water-based. We are all water, and we're assembled in water in our mother's womb. So how do you design in digital conditions and with digital machines with water? Most of the industry is not ready for that. So we worked on this platform, and what you see on the screen is a sculpture made only of a few wood pedestals and chitin from shrimp shell, this very strong structural polysaccharide that comes from shrimp shell waste. So we demonstrated that we could do that it could be big, it could be interesting, it could be self-supporting, it could be digitally engineered. Next. So at the time, Jenny had figured out a way to propose an MRS Material Research Society panel by architects for designers, engineers, and biologists, and she called it Adaptive Architecture and Programmable Matter. And she mentored my partner and I to go there. And for the first time, you're an architect talking to material scientists, and it felt that it was possible. So we are super grateful for that. And next, that platform brought me to talk to scientists and to be able to interface with biomedical engineering for 4 years during my PhD, where I at the Tufts Biomedical Silk Lab, where they look at, what for them, a biomaterial is something you implant in the human body and it's going to heal or deliver a drug or allow for a cell culture to grow. So for them, a biomaterial has the possibility to heal. It's a device, it's something that we digest. And how can all of that help bind or build architecture at the same time, help it interact with the environment and degrade at the end of life? So that's why Dumalab takes this approach of radical, benign materials, and we don't allow ourselves to use PLA, we don't allow ourselves to use metal in the joints, which makes our life very difficult. And these are the brains behind it. But it's a good challenge to undertake, right? Can we do everything with natural methods? Next, thank you. And all of that work ended up being assembled in a Guauja, next, that Rick and Athena, helped us realize in Boston. And is now came back to San Francisco MoMA and, degraded. And here, of course, we took a shortcut. As you can see, the white never degraded. So, PLA, no way. So, thank you. This is Dumalab now. It's been 3 years at Penn. And this was Orkan Talent's, you guys know him, old biolab that we turned into a mashup of an industrial kitchen with robots and manufacturing, but also a lot of chemistry. Because what we do when we try to find interactivity and adaptation is mostly chemistry. We also, again, no one wheels around that we can make our life easier with. So my students come from biology and material science and architecture, and sometimes we have educators, anybody interested in biomaterials joins us and tries to develop what they think biomaterials can be. You see, this is the premise. We work with a biological concept and scale it up to tools that can handle it and that can assemble it digitally, so that then robots can deal with it and economically, robustly assemble those structures into architecture. These are some results that come up from the lab. One is a rock solid sand mixture that behaves like lightweight concrete, but only with a couple of centimeters and a stiffened by fiber instead of rebar and Portland. The next one is this giant cannon wig we built in the Land Design Week that reacts to UVI radiation and tells you about threat to your skin and your eyes. That you see also in the exhibit. My students have been very, very obsessed with waste material that we can turn into architectures that degrade at a certain programmed time. And what you see here is avocado pit or skins from fruits that you cannot metabolize, turned into more materials that can be canopies, that can be stools. And the last one is wood without glues, without epoxies, without formaldehyde, that can make lattices, that can make particle board. And you'll see in the exhibit a few stools that replace particle board just with algae gum and straw. So the journey is water-based, as you realize. Also quite radical because it's all assembled in room conditions and as less energy as we can. And I'll leave it here so we can have a conversation.
Okay, go to the panel. Okay. Okay, great. Thanks, guys. So I think now we'll have time for a quick discussion. The first question that I was wondering as I was listening to you guys is, what really is a biomaterial? Can you define that? Because don't technically all materials come from Earth? So what is that? Yeah. Oh yeah. Well, this question wasn't directed at anybody in particular. start, Leah, if you want, but yeah.
Yes, hi. Yes. So to us, Tumula biomaterial is something that we can digest and Earth can digest. So differently from a scientist that will say, okay, titanium, biomaterial is biocompatible. We say no, it has to be digestible, like you said here.
Thanks. So digestible and time scale is important there too, right?
We'll just go down the line here. So last night I presented biomaterials as a paradigm, starting with learning from nature, so a way of thinking, the way of thinking specifically with environment. So I presented the concept of reciprocity, which I learned early on from Peter and his team, which continues in my current collaborations with plant biologists and biomedical engineers, where we're looking at robustness related to morphogenesis, the role of mechanics in environment and specifying form, function, structure, etc. And then mimicking nature. I presented some projects in collaboration very early on with Xu Yang and more recently with Dan Lau. So literally extracting principles and rules from nature and then applying them towards the production, the design, and engineering of entirely new programmable materials. And then the third domain within biomaterials as a paradigm, collaborating with nature. And that's in the context of our work, where we're working with living materials, whether that's DNA glazes to selectively steer particular behaviors and responses to the environment, or more recently working with baker's yeast to metabolize formaldehyde in agricultural environments. And I didn't present that last night, it's not published yet, but we've had a lot of success with these really simple materials on ceramic substrates as a possible solution for some of these issues. So that's how we approach the paradigm of biomaterials.
Yeah, I love both of the previous answers, especially Leia's answer about something that the Earth can digest. I've been really interested for a long time in thinking about materials that do a job and then disappear gracefully and go back to component parts that can be used for things. Basically things that are mortal, that have a lifespan. That's the key to life. Most life has a finite lifespan. And so I find that extremely appealing. So that we don't end up left with giant concrete and steel hullets and sort of non-digestible plastics and others. I also have a kind of a personally a more multi-scale view of biomaterials. Basically, everyone in this room is made of a collection of biomaterials, or a collection of biomaterials. And we can think of them from the scale of the nanometer all the way out to the colossal, as those different scales as being relevant. And at each scale, there are different problems and different questions and different uses. And so about 35 years ago, someone at UCSF, a group of people had a very bright idea, and that was that the disciplinary boundaries between certain key fields, biochemistry, cell biology, genetics, and developmental biology, were artificial. And so they merged them into one graduate program called the Tetrad. And the reason for that is, if you think of the history of genetics, back in the 1800s, a monk working with peas came up with some rules for how traits spread through. They kept the word gene. There was a gene underlined it. The gene was a logical construct. And then over the course of the next 60, 70 years, one of the great breakthroughs in biology was something called the chromosome theory of inheritance. The notion that this gene, first off, it behaved as if it had some spatial component, and then it was tied to an actual material, something called a chromosome. And I'll let you know, it's just a blob of something that absorbs stain. And so there is a materiality to this concept. So a geneticist, they do a screen, they identify genes, guess what? Those turn out to be molecules. And so you have to think about the materiality of this abstract concept. And so people can't just study genetics, they also have to study biochemistry and cell biology. And cell biology is really the science of how molecules interact in space and time to create something three-dimensional. And so I teach an all-quarter course to the tech grad students on basically these concepts called macromolecules. It's physical biochemistry, materiality of life. And so that gene is a piece of DNA, it has mechanical properties. Those mechanical properties are absolutely essential to understand how the function of that gene is regulated. Camille's project, for example, is to study not just how the materiality of DNA controls the way DNA is turned into proteins and molecules of life, but how large swaths of DNA are packaged and how they create what are called condensates, and how it turns out the mechanical properties of the nucleus are extremely important for determining the fate of a cell. Stem cells that can turn into many different types of cells. For many years, we thought there were chemicals that were the key determinants of them, but it turns out that stem cells that have not committed to being any type of cell, what they do is they feel their environment, they tug on it, and they measure the mechanical properties. And if it's soft, if they're in a squishy environment, they're like, must be the brain, I'm gonna turn into a brain cell. If they measure something that's sort of intermediate, they're like, oh, it must be in the muscle, I'm gonna turn into a muscle cell. If they measure something that's very stiff, and again, no chemical cues, just the material properties around them. It's very stiff, I'm like, oh, I'm in a bone, I'm gonna turn into an osteoblast or osteoblast. So cells measure mechanical properties, they care about them. If you think about how we detect cancer, often what do you feel? You feel a lump. What is a lump? It's a group of cells that have decided, I don't care to match the mechanical properties of the cells around me, I'm gonna do my own thing. So I feel different, the cells feel different. So biomaterials, this is not to say anything different from what other people are saying. I'm just saying, when I think about them, it expands down into the micro scale, into the cellular scale. And what I'm really interested in is the rules by which molecules assemble in order to create these different processes, right? Create these different properties, and how those can be regulated. So if you ask me what is a biomaterial, I would say it has to do with, personally, it would have to do with evolution. Something that's rooted in the evolution of life as it exists now.
So for me, coming from architecture, the biomaterials is defined by where it comes from, where it's sourced. So in a way, biologically derived materials for me are biomaterials, and that's a very broad category. It's almost like a super category that then trickle down into other topics. But I think the fact that it comes from organisms or is extracted from organisms is what defines it now. In its best sense, yes, it sort of can return to Earth and can be thought of not just by what it is made of, but also by the processes that make it, right? So I think designers are now engaging more with using waste streams, let's say, into biomaterials, as opposed to extracting, from raw or endangered sort of species that in design was, also kind of held in importance. But I think biomaterials is very much like this very big category under which others exist, such as the engineered building materials. I think the main difference there is that there's like a friction with biomaterials, which is that they are derived from organic sources, which makes them not as consistent, let's say, as materials that are produced chemically or sort of synthetically or mechanically. And there's this tension that we want them to act statically, to have very defined properties that we can sort of rely on and design with, whether that be for architectures or products or structures. With engineered living materials, that sort of gets even more complicated in a way, because now we're interested in retaining those living properties. And as I was mentioning before, you start to blur the boundaries between material and organism, and I find that incredibly interesting. I think it's maybe because I am naive, but I really want to engage with the fact that it's living. and to think about that as a design problem. So embedding living cells, particularly bacteria, that's my sort of focus, into material substrates for, let's say, regeneration, self-repairing, sensing, and remediating, adding other functionalities. I think that can help us address environmental issues, but also maybe think about experience as well. So what are the smells, right, that these organisms can produce, or compounds that can perhaps help us, whether that be with focus or relaxation, or creating environments. I mean, in design and architecture, there's light, right? We think about, healthy spaces that make us feel good. So how is this another layer onto material that helps us address the health of our environment, so at a deeper scale or a smaller scale, perhaps. And so the engineered living materials for me are within biomaterials, but then they seek to retain the biological processes, the biological metabolism for added functionality. So again, it's very, very broad, but I think it presents these other interesting niches that are left to explore. So the engineered living materials, I think, is where I would like to take my work and my research and think about incorporating them into materials that are biocompatible. So now all of a sudden it's not just thinking about materials that, let's say, withstand mechanical properties of interest, but also that are bioreceptive to light. And that's also incredibly interesting, right? Because then I think about our culture around plants a lot. That we can have plants in the home in quite perhaps the same way that we do today, where early in the 19th century, and it comes from colonialism, but this idea of creating like Edwardian cases or structures that would support life over time, right, and bring them to different places. How can we take those principles, but instead of perhaps being extractive, think about it to create more empathy or different relationships with the organisms that we inhabit? So now we have plant moms and plant dads, and that's sort of like a badge that people wear with pride. And so I think embedding organisms, specifically bacteria or cells, into materials feels challenging because we think about sort of dissociating them. We want them to be self-sufficient, self-reliant. But I think there's an opportunity there for design to engage, to think about their constraints, and to imagine new experiences. Going back to design and practice, like I think for me, working with these materials and these processes in a very scientific environment was daunting. And I think at the end you're like, am I a scientist? Am I doing these biomaterials? But we should also think about them through the lens of design, as many of us are seeing today, and see biomaterials as not just what composes them, but also the processes that are involved in making them, and then perhaps in the future implementing them.
Thank you. Yeah, just anecdotally, I can add that what I liked as a biochemist working on the biodesign team was that with biomaterials, we started really with the material and characterizing it in a detailed way, like on a molecular level, testing how resistant it was to various forces. And then from there, we were able to kind of characterize it among other known materials and decide what's the best form for this material. And that was really mind-blowing to me because my naive understanding of architecture was that it was very formally based, and you would start with a form that you wanted and then kind of fit the materials from there. And so it was great to see how creative and how much room for creativity there is in biodesign. So that brings me to my next question, which is, what are some effective models for collaboration between disciplines in biodesign? So between scientists, like molecular biologists, and architects and artists. Yeah, if you could speak on that. Anybody can start. Yes, yeah.
Should we go this
Lauren, you can start.
So, I mean, that's such a great question. I think for me, communication through prototyping and imagery. So often, I sort of read up enough and then try to produce prototypes, and then, we get a little hint, I mean, you know, I eventually got to bricks, and that was like incredible. It took like 2 or 3 years of work. But when I got the first crystals, I was like going around to scientists, and I was like, can we image this? What do you think about this, right? And like when they see that, and when they see the vision, it's like, okay, let's talk about this. Okay, how did you get there? You know, what did you do? And so I think as designers, we have an incredible tool set to communicate, to share ideas, to share the visions of where we're going, and that's so powerful. And so a really big part of collaborating for me is to bring others in and to show them something they can feel, something they can see, like where are we headed with this? Oftentimes that takes a lot of like mucking about in the background and trying to figure things out. But I think it's a wonderful way to share excitement, just making, iterating as a process to collaborate.
I would say the most important thing is to hang out at the cafe at the local art school and look for people who look like they might want to talk to you. I think it's all about people. I think the thing that's hard for some people to understand about science is it is a social enterprise. It's all about, science is all about intersocial interactions, how well you get along with people, who you get along with, how are you thinking about a problem in the same way as someone else, or more importantly, are you thinking about it in a different way? Can you do your differences sum up to something bigger than what either of you could have done by yourselves, or are you talking across each other? So I think finding the people on the other side of whatever divide you want to bridge that also want to bridge that, even if they speak a different language, maybe especially if they do, is the key to doing collaboration, not being afraid to do it. I was asked before this started about how do you talk about biology to artists? And I said, well, they don't understand how you dumb it down. I don't think it's about dumbing it down. It's about talking about it in absolutely the most accurate terms. Now, maybe we speak different languages, but I think I found that the architecture students, artists, have no qualms about asking a question, what might be a dumb question, because it's not their field. They have no ego invested in it. I would have a terrible time asking a question that's in my field that I thought was, oh God, I should know this. But outside of my field, I have no problem with that. So almost the farther apart you are, the easier it is to communicate in some ways. The easier it is for me to ask a stupid question to an architect. Like, I know this is a dumb question. You guys probably think about this all day. I didn't think building sales. Things like that. So I think that recognizing that there might be other people out there who want to bridge this divide and finding those people. Now, the interesting thing, there's an essay by Lewis Thomas called The Woods Hole Cantata, which is sort of a smug essay about how scientists are interested in art. And it's about this group of scientists every year at the Marine Biology Lab at Woods Hole who get together and sing Bach cantatas. And it's about, and it has the line of, I doubt that artists ever get together and read Darwin, So that's about how scientists really do appreciate art. But my God, artists are really interested in science, and that's something that a lot of scientists don't realize. And so I think finding the people who are interested in an honest way in bridging that divide and just engaging and continuing to engage, teach each other your language or find some sort of intermediate common language that you can both speak. I think that's the way to go.
I couldn't agree more that successful collaborations are fundamentally about the people. It's about respect. It's about clear communication. It's about asking those dumb questions where you feel they're dumb when usually they're not. They usually spark really awesome discussions, but not being afraid to voice that. And a commitment to hard work, a shared commitment to being in it for the long haul. And Aaron, Margaret, you mentioned this, once you find that collaborator where the spark is just the super sauce, you don't let that go. And I now have collaborators, as I mentioned last night, and there are others that I've worked with for many years. And it's also about laughter. It's so much about successful failures. It's about, taking time to play and get out of the lab or the studio. Peter and I wrote a book called Lab Studio, and we used to joke that we could write a second book called The B-Sides. The stuff that we'll probably, never talk about. But the other thing too is like, you find your people, and it took me a while to establish my networks at Cornell, and we've now found the unicorns, and it's so exciting to be in that type of space. And maybe one other thing I'll mention is we were successful in procuring one of the NSF Convergence Accelerator grants for phase 1, which if you haven't looked into the Accelerator Convergence grant opportunities, it's a really great space for this type of work because it's focused on application for societal impact and like broad range of project topics. But there's a curriculum that you go through. I felt like I was back in graduate school, super intense. But one of the core parts of the curriculum is called Team Science. And it's focused on creating a space for having open discussions around the kind of psychology of collaborating together across disciplines. And that was a really transformative experience for me because it just gave the space and time to ask tough questions around sort of relationships, things like, when you say that really bothers me. And to be okay with that, or to say, man, the way you run your team is just so inspiring. I've learned X, Y, and Z. And so that's been a big takeaway for me is effective collaborations and producing amazing results that you publish in design, in science, etc. is so much about that effective communication and space for working together successfully. So I really encourage all.
We thought microplastics were bad, nano cadmium crystals or whatever, would be even worse. So I think that it's aspirational, but I think it has to be something that we would be safe eating, or that the earth can eat, to steal a phrase.
I love your vision, and I think to realize that vision, we would need a massive paradigm shift. And part of that is technical. And one of the things that we learned in the Materials Research Society Symposium, this was years ago now, 10, yeah. We need to publish a book. You reminded me. I didn't call them. One of the things that we realized, we had an amazing group of architects and scientists, Joanna Eisenberg, yourself, Skylar Tibbetts, and just an amazing group. But by the end of the second day, what we realized was that there were a number of people there that were missing. We needed people from law. We needed people from business. There are litigious issues around building code. They go back to the 60s. That these hurdles are real in terms of actually getting what we're doing in the labs into the built environment. So there are those aspects. And then, a paradigm shift in the way that we think about sustainability. Like, we're still in a paradigm where it's focused on resource consumption, and I'm not saying this room, but a shift into building for not just our generation, but for 7 generations down the road, and that will, I think, take time and shift away from our reliance upon petrochemical materials. So I think we all are aspiring to that vision. That's what keeps us going. That's what keeps us going after the grants. What keeps us up at night and excited with our students. But I think over the years I've realized the very real hurdles that exist that are outside of our domains, that if we can't connect those dots and bring those folks to the table, it will be hard. But I don't think we should ever stop dreaming and keeping that vision alive. And maybe the last thing I'll say is that I do think it will be a hybrid, right? In terms of the reality of living architecture and what that could be. both static and living and changing as a kind of cohesive, integrated set of relationships.
Yeah, the sci-fi is the house that grows itself, right? And it's going to be wet, and it's going to be squishy for a while. It's going to be tender, and right? And do we want that? We might. Actually, Mitchell Joachim just assembled a few willows into a braided hut and demonstrated that it can be a living house that he's been living since and finally it's happening, right? But of course we don't know where that's going. How do we inhabit a living house that's made of trees and it has many other organisms living in it? The most close or lowest hanging fruit here is bioreceptivity, right? You treat your skin as a bioreceptive system. And it's all about, concrete that has the right pH and the right porosity to welcome mosses to grow on its skin. Or, we're working on some portable bricks that the moment they touch the ground, they bioremediate and trap heavy metals and then are able to grow healthy food on the surface. So it's coming, and it's for now, I think, we are all looking at the skin. Because of course, at the end, we don't want wet architecture, right? We are starting to welcome the biome in the home, right? And Laura knows this very well. We're starting to be okay with, there's other organisms in the home, and how do we deal with this microbiome inside? But I don't think we're ready for wetness. We want soon. But also wetness, and you mentioned water, is going to be there for biodesign. And talking about, I know this is boring, but talking about the ASTM standards, the way we characterize everything we make, all of us, it has to change. Because you're doing fire tests, a corner fire test, and you're doing compression and tension and bending and abrasion tests, but you're not doing regenerative index tests or water swelling in the presence of humidity or morphing because of X or changing color with light tests, which is all of the enabling that the biomaterials can bring to the table of architecture and product design. So we lose in the current ASTM standards, but we'll win when we change them and the code changes for those grown homes to happen. But it's going to change. We cannot reuse injection molding if we are working with materials that need to evaporate that water out of them, right? We cannot close them. So when, and I'll shut up in a second, when Coca-Cola comes and says, how many bottles can you make in a second with your biopolymers? And you tell them, well, it's like 14 hours of the night. And it's flat, water-based, so then I need to fold it into origami, you 2D to 3D, they're like, what? But it will come, we will solve it. It's going to be an industrially produced system. So just the industry today doesn't work.
Just really quick, just to end. Your question is why I got into all of this, and I think for me, my background in architecture, that was the dream, that was the vision, and I actually really want to hear Martin's answer on this, maybe during his convergence, because his work is really inspirational for me, diving into MICP or bacterial biocement, because it's the most tangible way to get there. And I think one of the discoveries that I really loved was that the system continues living beyond when you formulate it. So for me in my research, I wasn't just interested in producing one tile, but many tiles, and actually if you keep feeding them, they grow and they bind together. And so it was a way to continue to scale and scale up. I think the biggest hurdle is what Lana was mentioning, time. Time to grow these, time to cultivate. We would have to have so much patience, I think, as a society, and foresight to thinking into generations future to start to welcome that. And we will get there. I also believe that. But I think it's going to keep taking more collaborations and more time. But yes, your question is so important, and I think will inspire and continues to inspire a lot of people to get into biodesign and biomaterials in the future.
So this is just a quick comment on that same question, Simone. I was working, as Jenny said, I sat in on her class back in 2008 and was scared to death to be her student. I ended up writing 10 years later, owing to a deadline that forced me to write because I was still terrified, the book Toward a Living Architecture that Minnesota published in 2018. And it addresses throughout the whole book kind of your question. It was a driving factor that I was actually serving as a critic of rather than a proponent. And so I think I called to task some of the, I'll call it somewhat pseudoscience stuff that was happening, at least in the 2010 to 2015 period, which was my data gathering phase. You know, the book now might be dated. So I just have to say, if you're curious about a more critical view of it, you might look at that historically as insightful. But on the other hand, I have to say, things have changed in this community. When I first started doing this research in 2007, I felt like I was very alone, except like I found you and Peter, or then I found a few other people. But for the most part, the ones who were doing the good science that I felt that I respected in collaborating with architecture and design, it was fewer and far between than it is today. And honestly, I'm so excited to be here. I was really shocked yesterday. I actually started crying when you started talking, Jenny, because like things have changed in 15 years. We now have really good scientists and really good architects and designers who are doing really thoughtful and critical work. And things feel more possible in a more careful way than they did to me when I first started writing that book. So I don't know if I buy the vision about, could we, I think if I heard you, the idea is programming the DNA in the organism to build the building itself for us. In other words, you're controlling to the utmost degree the outputs of an environment produced by an organism. That to me seems really audacious and controlling and not necessarily the paradigm of collaboration I see. I could be misinterpreting it, but that's part of the criticism that I have throughout the book is like, to what level are we collaborating or are we trying to control? And also what's scientifically feasible across scales? And those are really complicated questions. So yeah, here to all the people who are thinking about this and being motivated by it. But also I think thinking ethically and carefully about what the implications are. Thank you.
I think there's one more question. We have time for maybe response.
I'll be really fast. Thank you. I've learned so much from your panel. It's been really enjoyable. I was wondering if you could each offer an idea about addressing some of the challenges of taking your research into the real world. Like, what are some of the fun ways that you see that moving forward?
By real world, you mean that
commercialization, or however you see it becoming implemented in a real way outside of your lab?
Yeah, so maybe I'll address your question through a relatively recent collaboration with IMI, which is the International Masonry Institute, IMI. Amazing consortium of people, and I, wouldn't have thought that Masons were interested in like cutting edge research around bricks and masonry units. And so I gave a talk a few years ago and then have participated in a few other events with them. And so we've been talking about developing a demonstrator wall together with Masons who have extraordinary knowledge around how these systems come together, but to really innovate and learn from each other. So that's an exciting example of trying to push the prototypical work into potentially a material system that can meet the built environment. The other way I'll address your question is just the real power and role of demonstrator projects. And as I talked about last night, making sure that those projects reach broad and diverse communities and spark interest in the origins of the work, whether or not they understand it doesn't matter really, just that it provokes some questions. And because that's, I think, I firmly believe that those impacts are how we will realize the translation into the built environment. Kind of large-scale buy-in and excitement around this work, because most people haven't seen this before, right? We have to help educate. And so the pavilions, the demonstrator projects, the exhibition venues, the competitions that we go after are equally important to the fundamental research in terms of educating broader audiences.
Right, and I do think that we might not have all the parts. I was talking to Alan yesterday, and he's saying, I have a core of a wall. Do you have a scheme? Right? So it could be that we get there by layers of each other's work, and you can solve everything. We are looking at, okay, so how do we waterproof without fuel? But can we make things water resistant? And then someone has been looking at this with in the leather industry, et cetera, et cetera. And Alan has been looking, professionals have been looking at making very good core structures, but now how do we finish them, right? So we might have found a way to make hyper-sustainable particle board. Well, that's when I think this might become a system that then it can be commercialized in the real world. I mean, I don't think I would work in the real world. But as you are certified, right? Certified real world. Yes.
This is slightly, well, it's not really off the topic of biomaterials, but it's, I recently got interested in human-made closed ecosystems. Now, there's not a lot of research done. Most of the research was done by Soviet scientists in the 1970s, and a lot of that's only recently made into English. And it was only in the 1980s that people started to figure out how to take collections of organisms and put them together, seal them in a tube, give them a little sunlight, and have them live together forever with no extra input of energy. The reason why I'm interested in that is because the Earth is moving more and more toward being a human-made closed ecosystem. As species diversity goes down, nobody wants this to happen, but this is what is happening. And so we have to understand what it takes to have a closed, sustainable ecosystem. When are we getting near the catastrophe points? When are things, and what does every new technology, every new perturbation, every new way of doing things, how does that help us or push us in the wrong direction? So I think, for me, understand this is why evolutionary cell biology sounds like a very academic topic, but I think it's really fundamental to understanding how the Earth works as a originally wild, self-assembled, evolved ecosystem that is becoming much more Anthropocene.
And then for me, with the bacterial biocement, there's a few issues. Primarily durability. So far, we've only worked with bacteria, salmon aggregates. We've tried different additives and mixtures as well. We've never sealed them, and they hold quite. They're robust. I think during these workshops, it's also been a great opportunity for me to learn how they perform long term, because the students, they get to take them home with them, and they're in these conditions. And so I have them sometimes reaching out to me saying, oh, it's like been a year, and Laura, it just crumbled. I didn't even touch it, it just crumbled. It's like, okay, interesting, we gotta figure out why that happened, what were the environmental scenarios. So there's durability. I think there's also issues of containment, and we were trying to work on this with the work that we were doing at GenSpace. I think primarily because the way we treat the bricks at the end is we put them in an oven to sort of eliminate the microbial activity. They form spores, and they're actually quite robust. And so how can we guarantee that these spores aren't going to get into the environment? How do we think about containment? So part of the work that we were doing at GenSpace was not only use local soils, but then also start sequencing local microbes. Does this sort of perception or the way we think about it change if we use local resources and local organisms? So if they do come back into the environment, well, they were there to begin with. And so I think those are two primary challenges, and then we've had inquiries about scaling up or doing a wall. And then you just run into, well, I'm working in a biolab. And so there's issues of space. You know, a lot of the work is for biological engineering, so you're working with 20 milliliters, 10 milliliters less, oftentimes 1.5 milliliters. And all of a sudden I'm coming in and I'm like, oh, I need 10 liters of this media to feed all of the bricks, you know, that are going to go into this production. Oh, we need bigger equipment. We need to change the layout of the lab, change the layout of the work in the lab. And for a community space, that's not, you know, the sort of way that we work, because there's a lot of other research, important research going on. And so how do we mitigate contamination across species, across organisms, and also work at scale? Those are the hurdles right now that we're facing. Again, we can, you know, kind of work through them, but I think right now that's the immediate hurdles.
Okay, well, thanks again for our panelists. Yeah, lunch break. We'll come back at 1 for anybody who hadn't heard.
Take that space equally seriously as you do the doing of the work. Because I've been in some bad collaboration. I'm no longer working with those people. So it's, those spaces are important to sort of take care of yourself too.
I do agree about the unicorns. They are so special. Any engineer or scientist that appreciates design, or that, you know, thinks, not only thinks design has a say in the table, which is quite advanced, but that has had a contact with art in their past, with any creative endeavor, will be much more prone to understand and learn and collaborate. And these unicorns are hard to find, and again, I haven't stopped working with my Boston collaborators, because even if they are far now, it's just so important to find them. And at Penn, of course, Shurian is one of them. And still, you know, you have to be understanding that you're going to talk different languages, because when she introduces me to an NSF call, she'll be like, and now Laia is going to show pretty pictures. But what she means is their science, that it's beautiful, right? And it's beautiful. So she is very engaged like that, and I think that's why we all put demonstrators out there, and it's this pavilion that we're calling it, right? That is just a demonstrator for what can be and for scale. And when Javier and Don at the Lease Institute saw the ocean, you know, the giant chitin structure, couldn't believe, because they had been training us for like 2-3 weeks on how to process chitin and how to get to different molecular weights, how does it self-assemble, how it works in dry, because, you know, in science it's wet. And when they saw that, they believed. And Javier took a job at SUTD and, you know, partnered with the Stelios and built the biggest cellulose and chitin structure there was, because he was so inspired that this could take scale. So scale and the unicorns, and then also I think, and I tell my students all the time, it's about standing in the shoulders of giants. So truly understanding everybody else's research and coming with, yes, maybe the naive question, because we are good at naive questions, but with so much scouting and so much understanding of where the others are at, and that respect does become very, very productive, and we've experienced that in the lab many times. So for sure.
Okay, thanks again. Yeah, let's give another round of applause. So, work like this is obviously very resource intensive, time intensive, expensive. How do you get access to the necessary materials and money for this within academia, but also outside of academia? And Laura, I thought we could start with you, just because you're in a unique space. Yeah, we talk about that.
it's such a great question, and I think for me also, because I'm fairly early stage in my career, it's been a bit challenging to find funding. So the way that I've navigated it is through finding collaborators and through applying to a lot of grants. I found the arts space to be actually really welcoming to this type of work, because so integral to it is not just working it out in the lab, but then doing these parts, these prototypes, these sculptures that start to scale it up, start to show the possibility of where the work can go, and that has been supported by arts grants for me independently, which I've been so fortunate to receive. I also collaborate with wonderful institutions. GenSpace has been so supportive, not only in providing space, but also in helping to fund some of the research. And what I've gotten progressively better and better at is doing this work with very minimal budget. So I've learned new techniques, switching out, what is the sort of minimal viable version of what I'm trying to do? How can I prove this so that I can go for bigger grants and sort of start to scale up? And I think maybe going back to the previous questions, collaborators are key to this as well. I love teaching workshops, not just because it helps make the information more accessible, but I meet incredible people that have incredible ideas and projects. And one of the ones that stood out to me was I was recently teaching a GenSpace workshop, and we had a beekeeper join. And he produces these beehives in New York City, and they're made with aerocrete. So it's these bricks, essentially, these big blocks that are made of concrete, aerated. And he was saying, hey Laura, I attended your workshop, I don't know anything about biology, I'm not going to pretend to now try to do this myself, but would you be interested in collaborating with me to do a beehive in a community garden in Harlem? And I was like, yes, let's figure out how we make this happen. So together we've been applying for funding, getting help from the Harlem Community Garden as well as GenSpace and other sources to try to make this possible. With that comes issues of, okay, now we have to tackle scale. But I definitely wouldn't embark on it by myself. And it's such a wonderful way to think about the different applications of the process, paths that you didn't anticipate. Because what I love about the bees as well is that it's not just about replacing the material, but he also told me this wonderful story about, he was really nervous about the smell. You guys have never smelled bacterial biosimilar or MICP, it's maybe more technical term for it, it smells. Because it releases ammonia. Some say it's like a sweaty sneaker or a kitty litter. It's not very nice. But he was interested because his grandfather would tell him that his grandfather would tell him to go pee by the tree by the hives. Maybe there's some ties to urea, to this ammonium smell that they actually prefer. So all of a sudden, all these ideas are coming together, and we're finding ways to fund it. So I'm incredibly grateful for arts grants and the institutions that I'm a part of, whether that be GenSpace and also Cornell, to support some of that early work and just getting good at making these really tight budgets. So that's been my experience so far.
That's a great answer. I think there are 4 maybe answers to this question, or 3. We'll see how many. One is being very creative and resourceful, independent of any support you're getting anywhere else, and that crosses all sorts of boundaries. What I mean by that is, in science, there's this and being generous to other people and creating community. So in science, there's this movement for open source science, open source information. If you, and many of us now, rather than waiting 2 years to try and get our paper published in some fancy journal, we immediately put it on Biomaterials, put the preprint up, get the science out years before it would otherwise get out. Everyone benefits from that. And there are people who are like, oh, but I've got other things I want, but no, just get it out there. Share. So if you come up with something, figure out how we can build a community where we share information, ways to do things, approaches to materials. And part of that, or related to that, is something that I think that Margaret and Evan did that was brilliant a few years back during COVID, when they were running a class remotely, where people were all over the world. Some people weren't even allowed to travel to the US, but they were in this class. And so I had this brilliant idea of making the class about materials and about whatever materials you have at hand. And as Margaret reminded me, one person was in China, they were in lockdown, all they had were, well, I have some eggshells and chicken bones. And so what can you do with that? Can you make a biomaterial out of that? Can you create something? So, you know, if you have no resources, look around. Maybe there's something around you, maybe the eggshells that come from all the restaurants in San Francisco can be leveraged to build something, for example, to call back to this recent design challenge project. So that's one thing. So, you know, being creative, using whatever is at hand and sharing in a landscape where resources might be scarce. The second is, I think we need government funding, right? And you know, what is government funding? It's what we have all decided that we, as a culture, that we want to fund. It doesn't come from the largesse of billionaires. It doesn't come from, you know, any immediate commercial application. It's, as a country, we have, you know, decided that it's important to invest in certain things. So we need to protect and create state funding, local funding, federal funding. It's as a, you know, as our kind of collective will to push this forward. So I think that has to be an essential part of it. And then on top of that, there are private foundations. Obviously, that's extremely important. Most of the work in my lab is funded by a private nonprofit foundation. And then the last are industry partnerships. So, you know, we want to get this work out into the world. We want it to have practical uses. We want it, you know, architects want to be able to, you know, draw from a catalog of materials and new ideas. So I think those are the four things. One is kind of a grassroots, you know, rowdy, open source community. The other is kind of institutional government funding, and then private foundations, and that all of them have to be working to have a healthy ecosystem. And just put a plug for, we were talking about GenSpace in New York. Some of you may know in the Bay Area, there's counterculture labs over in Oakland, which is a similar kind of biomaker space, which is kind of fun thing.
So in the early years of my work, it was super important to exhibit the collaborative work in venues outside of each other's discipline. We were successful in showcasing some of the early spatial prototypes and installations, material systems at SIGGRAPH in 2008 and 2009 in their design and computation galleries. We entered the visualization challenge that AAAS puts out and won first place. This was for branching morphogenesis, and so that project ended up on the cover of Science, which these sort of pivotal moments, like suddenly everybody's like, oh, what are they doing in Lab Studio? And I owe so much to my scientific colleagues for showing me the ropes in terms of what it takes to successfully procure an NSF grant, for example. I mean, as someone coming from architecture, $2 million was like, and I mean, to the scientists and engineers on the team, they're like, well, this is a small grant. This is kind of just so, you know, it's like the sort of different cultures that we come from. The other thing I learned early on is that you don't get NSF funding without prior NSF funding. So you align yourself with collaborators and learn from them who have been successful. And also I learned that, like anything else, the NSF is an institution. It's made of people, and even if you're not successful with a proposal, it's always worth the time and effort to talk to the program officer, to form relationships, you know, to put in proposals that maybe they're not even soliciting to see if there's funding available, which is how Maria Pazzo-Charles and I were able to host the MSR, that symposium early on. And so we, over the years, have been successful primarily through the NSF, the sponsored research. We've been quite successful within their umbrella, which is called Emerging Frontiers in Research Innovation, which is the space where you can do weird stuff, really kind of out of the box thinking. They're really focused on convergence research and collaborations that cut across disciplines. We've also been successful with industry, and I mean the great thing about industry is that it's usually much quicker in terms of securing the funds versus going after sponsored research. So for example, I spent an amazing day with some colleagues from Autodesk. This was years ago, related to the precursor program to Design Tech, which was called Matter Design Computation. And we had an awesome day in my lab, and that evening they wrote back and said, we want to give you $300,000 to help fund the first students. I was like, just spent 6 hours together. So again, it's about people, right, at the end of the day. And then more recently, well, maybe I'll back up just a moment. So once you get your first NSF grant, then you're building up your portfolio, and that's really helpful as you move forward. And then more recently, we were successful with the NSF Convergence Accelerator grant opportunities within TIP, which is Technology, Innovations, and Partnerships. And the NSF added that directorate in 2019, and they had not added a directorate for over 30 years, which is kind of amazing. And they have put a lot of funds towards that in relationship to the other 6. So there are opportunities, as I mentioned last night, we were supposed to hear back about phase 2. Cross fingers. So it's an uncertain time for sure.
So funding is what keeps me up at night. Every night. And they know, because we've been writing so many grants this year. One thing that has surprised us, and that's been really, really successful lately, is that we realized that our work, like Laura was mentioning, it's so basic in the molecular level, in the structures that we work with, that can be sourced truly from non-pure, non-processed sources. So Bhavna has been traveling to the Southeast Asian market in Philadelphia and gathering all of this bagasse waste from sugarcane juicing. And we have a giant room full of free straw that's hyper-locally sourced, that we build those stools you saw there with and brought back to the market for the vendors to sit on. So it all was a mini circular economy that costs us nothing because our machines exist. And yeah, we had a couple of buckets of alginate around to bind it all together. So those waste materials that we are valorizing have been instrumental. We work with dyeing facilities at Penn. We talk to chitosan producers that instead of giving us ultra pure sigma or a Pfizer grade chitosan powder, just give us a bunch of shrimp pills and a couple of antennas and a little net. But we don't care because we're not implanting this into anybody. It's not like it's not an immune reaction that right? It's just one panel that has a bit of an antenna. So we're all so grateful for this low grade, packaging grade, architecture grade one day biomaterials that people are producing. Same with we established a partnership now with a poultry farm that cannot source back all the eggshells because there's so much you can put back into the system, and that is very exciting to turn that into building materials. I'm sure you guys know. So those waste, that waste that is such a bad word for such an amazing source, has been instrumental in because we are a materials lab, we need that all the time. The second thing we've realized that we've been quite surprised by is that because our body is so simple, so benign, and so healthy, it can influence very much young children. So we've been running a ton of ecological literacy workshops that are hands-on, that teach children how to work with technical materials that they can leak, right? So it's very exciting to them. They have the same aha moments that you all had when you turned on an LED in an Arduino, but in another world, which is pastes and making and seeing plastics that suddenly dissolve in a bucket of water, and then they look at the acrylic and what? So we've been excited by that, and then suddenly we realized, with talking with educators and sociologists and psychologists,
that this is child development. This is, child welfare. This is child resilience in the face of climate change, what you were doing. So suddenly we are now in a consortium self-grant looking at helping children all over the world with these practices when we thought we were, playing and having, almost, Reggio Emilia opportunities for them. No, it has other implications that when you talk to experts in other fields, you realize you're making an impact you didn't know you were making. So now, we're not only applying to manufacturing and materials grants, which sometimes we are at a disadvantage with engineers and scientists, but we are applying to child welfare and child resilience, and maybe sometimes some adult resilience haven't paid yet with adults. Anyhow, just this rhizome implications that your work has that you might not realize it has.
Okay, well, thanks so much to all of our panelists for such an interesting discussion. I think now we're going to open it up for some Q&A from the audience, if anybody has any questions. Oh, yeah, okay.
Hello everyone. My name is Simone. First of all, congrats to the organizers, the panelists, actually everyone that participated actively to this. It's great. I love interdisciplinary, multidisciplinary convergence. It's beautiful. My question is a bit of thought-provoking, maybe inspiring question. And let me just give you a little bit of a background of this question. So first of all, I am a scientist with multidisciplinary background, and I'm an avid sci-fi reader. So I guess that makes me as big nerd as anyone else in this room, which is great. So, and because of my background, I kind of have a dream, a vision about bioengineering, genetically modify microorganisms and bacteria in a way that we can actually build buildings or structures in a way that is codified in the microorganism, and perhaps these microorganisms are just going to use water and nitrogen and oxygen and carbon that is available in the atmosphere to build out structures. And these structures maybe are also self-healing. So now my question is, what do you think? Does this vision, this dream make any sense? Maybe in the far future we'll be able to do that? What's your prediction? But this is also a question for anyone else in the audience. Thank you. Is it too messy?
Well, personally, I love the idea of taking out a foil packet, ripping off the top, dumping on the ground, adding water, and having a house spring up. I think that is aspirational, and it's something that we've been interested in. We've been involved in synthetic biology initiatives in the past. We were part of Nanomedicine Development Center at UCSF, which was, the idea was to build next generation therapeutics. This is before the recent successes of, and actually some of the recent successes of the CAR T cells, these immunotherapeutics came out of that. We reprogrammed the T cells of a person and put them back into their body and then go and they fight cancer. We were interested, can we build artificial cells? Can we build platelets, artificial platelets, for example, that will quench blood flow? And then can we take that outside the body and build larger scale structures? I think we're quite a long way away from that, but that's the way biology works. It self-assembles. Now, in the macroscopic world, we don't see a lot of things that spontaneously self-assemble. Part of that is because we're not being bombarded by water molecules, like giant softball-sized water molecules that are hitting us at 1,400 miles an hour from every direction to make us kind of explore lots of configurations and bump into a gene. But the materials that we use don't do that. But I think it's pretty far away, but I like the idea of self-assembling, but again, kind and graceful materials. Now, the worst would be some sort of inorganic nanotechnology, some material that left nanoscale waste